Hi, I'm Sumita Roy, a scientist from the Wellcome Sanger Institute. Today, we are breaking down one of the most powerful tools in modern biology, polymerase chain reaction. It's a method that can turn a tiny amount of DNA into billions of copies. We will use a PCR tube where we'll make a mixture of the following ingredient: thermostable DNA polymerase, an enzyme from a heat-loving bacterium called Thermus aquaticus; magnesium ion, a cofactor the enzyme needs; buffer keeps the condition just right for the mixture; primers, short bit of DNA that tells the polymerase where to start; and the nucleotides, the A, T, C, and G, which are the building blocks of DNA. And finally, the template DNA, the bit we want to copy. Then the PCR tube goes into a machine called a thermocycler, which heats and cools the PCR mixture over and over again. Inside the tube, three simple steps repeat. Step 1 is denaturing. The heat breaks the bond on the DNA, causing it to break and unzip into two DNA strands. Step 2 is annealing. When the temperature drops, it allows the primer to bind to their matching sequences on the single-stranded DNA. Step 3 is extension. That's where the DNA polymerase gets to work. It binds nucleotide to the template DNA, forming new bonds and building fresh DNA from the original DNA strands. That's one cycle, and each cycle doubles the DNA. After 35 cycles, one piece of DNA can become over 34 billion pieces of DNA. Once the DNA has been amplified, scientists can look at the sample to check whether the specific DNA sequence they are searching for is present. PCR might seem a simple mix of heating, cooling, and enzymes, but it is a technique that transformed everything from medical diagnostic to conservation and forensic science, all from a tiny tube and a bit of clever biochemistry.